Right, in this lesson we're learning about transcription and translation, which is part of our unit with replication, but it is very, very different, so please don't confuse them. Here are our syllabus dot points. Again, it is only half of topic 2.7 and the rest are on our replication from last lesson. Right. Protein synthesis is also called transcription translation. So to transcribe something, it means to make a copy. To translate, we mean to change something into a readable form. So when we do that, what we are actually aiming to do is change DNA into RNA, all right, in the transcription phase, and then translate it into protein or a polypeptide. So transcription is the synthesis of the mRNA or a special kind of messenger RNA, and then it is copied and translated to form that polypeptide. So the synthesis of the polypeptide there. Right, there's so many pictures of transcription translation, but it is quite a complex process that can be boiled down to one little picture like this. So the very first step is so we've got to make an mRNA strand in here by copying a small section of the DNA inside the nucleus. So that's the instruction for making the polypeptide. The messenger RNA then leaves the nucleus, and then it moves out into the cytoplasm where it goes to a ribosome. The ribosomes can be free-floating or they can be attached to the endoplasmic reticulum. The ribosome is reading that RNA 3 bases at a time, called the codon, and then the ribosome actually drags in a complementary piece of transfer RNA, so another piece of RNA, to bind with it, and this is called the anticodon. And then an amino acid sequence can form because there is an amino acid attached to every single piece of transfer RNA. Quite a complex process. All right, let's start with transcription. So in transcription, genes on the DNA are sections which provide instructions for creating that protein. And the DNA template, which is going to be this purple strand at the top there, only one of those strands is used to build the RNA strand. And it's called messenger RNA because it's actually sending a message out to the cytoplasm to build that polypeptide. So another enzyme comes along called RNA polymerase, and just like DNA polymerase in replication, what it can do is it can drag in nucleotides for building the RNA strand that are complementary to the DNA template. So here's the template, and those bases start to join in. So we've got, obviously, A is complementary to T, and U is complementary to A in RNA. So TAC is called a start codon because it gives that enzyme a little cue to say this is where the gene starts. So most of those genes will begin with TAC as a sequence. So when this happens, the RNA polymerase moves down the gene until it gets to a stop codon. That's codon. So those codons are 3 bases long, and there are codons that are specifically coded for, hey, enzyme, this is where you end. So it's trying to copy a particular gene. So let's say it's trying to copy this section here. In order to do that, to be completely identical, we need to copy the other strand, right? And so the one we want is called the sense strand, and we copy it and transcribe it, to do that, we copy the anti-sense strand, or the opposite one. And you can see that this is TAC, and this will start with UAC, knowing that there's no T in RNA. So in transcription, once the mRNA has been created off the gene on the template strand, the DNA can recoil itself, and then the mRNA leaves the nucleus through a nuclear pore, and it moves into the cytoplasm to find those either free-floating or attached ribosomes on the endoplasmic reticulum. Now we're into that translation phase, okay? That we're translating that mRNA into a polypeptide. And weirdly, it's done using a ribosome, and a ribosome itself is a protein. So we've got a protein making other proteins. What happens is the mRNA, the messenger RNA, moves into the ribosome, and the ribosome actually reads the 3 bases at a time. So it reads that codon, and that codon is where it starts to translate into something useful it can use. For each 3-based codon, the ribosome finds and binds a complementary molecule of transfer RNA. So these squiggly things that are coming up on your screen, they're RNA strands, single-stranded RNAs that are folded in a particular way, and they're folded to expose a complementary sequence of 3 nucleotides, and that's called the anticodon. So the codons are on the mRNA, and they're being read down here, and the things flying in are the complementary anticodons on the tRNA, or the transfer RNA. Now, each strand of that squiggly-looking tRNA has an amino acid attached to it, so it can begin to build the polypeptide chain, you can see those little circles attached to the top of the tRNA. Right, so that one's got PRO, which is proline. That's a kind of amino acid. So as more tRNA molecules bind to the complementary codon on the mRNA, the amino acid chain starts to grow. And you can see that speeding up in this animation. They start to bond together, and then that polypeptide starts to form, and it can break off and go and fold itself up. So once a stop codon is reached, the amino acid chain is complete and it's folded specifically to form a new protein, so no more tRNA is needed. Now, we need to be able to predict polypeptide sequences from using a DNA strand. So here's our DNA template strand. If we are going to make sure that we can copy this part of it, we need to drag in some useful mRNA that is complementary. So the complementary mRNA strand for TAC to start with would be AUG, and then we've got this 3, so GCC would be CGG. We've got ATG, so this is where it gets like AC, and then AAT would be, what would AAT be? U, and A. So once we've got that, we have to go to our important, really important table here. And this translates the mRNA codons, 1, 2, 3, 1, 2, 3, 1, 2, 3, into our actual amino acids. So if we've got AUG, we go, all right, here's my first one, it's A. We'll go across to U, and we can see that this is the start codon. So we've just got the MET, which is our start. Then we've got a CGG. So we'll come up here and go C, and we'll go across to G. CGG is an ARG, which is probably arginine, I can't remember. And then we're going UAC, so we'll go to here, U across to AC, so TYR, I can't remember the name of that one, and UUA, here's U, and here's A, so LEU. So that's literally our amino acid sequence building our polypeptide. Now, the beauty of the genetic code is that it's universal, so organisms have a way to create proteins regardless of where that protein or that instruction has come from. So it can be manipulated for our benefit in bacteria and yeast. So E. coli can be used to make insulin for diabetics. So what we can do is we can transfer the insulin gene that we know codes for making the protein of insulin, and we can put it inside the circular bacteria DNA. Once it's inside that bacteria, the bacteria just does what it does, which is continue to make proteins, and it just keeps making the insulin protein for us. So we grow those bacteria up, they create the protein, and we can extract it all off and use it for our own gain. So here are our dot points. Remember, it's only half of topic 2.7, and it is lumped in with replication, but still a very complex process.